Okay, so this is a revision video on nerve impulses. So there are 7 questions which cover the key pieces of knowledge you need to know for this topic. So pause the video, try the questions, and then you can play the rest of the video to mark your answers. Okay, so first question, how is a resting potential established across the neuron membrane? So I'm going to show the animation, and as we go through the animation, the marking points will appear so you can mark your work as we go. So first 2 marking points are that the sodium-potassium pump is actively transporting 3 sodium ions out of the axon for every 2 potassium ions that are moved in. And then marking point 3, you need to mention that potassium ions then diffuse out of the axon down their electrochemical gradient. Fine to just say their concentration gradient by facilitated diffusion through these potassium ion channels. They are not voltage gated, they are just open channels. They are also known as leak channels because they are always open, allowing potassium ions to diffuse out. Okay, let's keep going. So marking point 4, you need to state that sodium ions cannot diffuse back into the axon because voltage-gated sodium ion channels are closed. Or you need to have expressed the idea that the membrane is more permeable to potassium ions than it is to sodium ions. Remember that the membrane is not going to be 100% perfect in terms of stopping sodium ions diffusing back through. So some will leak, potentially through other channels. But the key thing is that more potassium ions are diffusing back out than sodium ions are able to diffuse in. And then finally, this gives an overall negative charge on the inside of the axon. That is the resting potential. So we said it's minus 65 millivolts here. That can vary, so don't be too fixed on a particular potential difference. But for this example, we'll use minus 65. Okay, that's resting potential, so let's move on. So now we're going to go through the answer for membrane depolarisation, how an action potential is established. In this example, remember that the sodium-potassium pump and those potassium ion leak channels are still in the membrane. They're still present, but we're just focusing on different ion channels for this. And also on the right side of the screen, I'm showing a graph of a change in the axon membrane potential, or the neuron membrane potential. So this is going to help us track what's happening here. So we'll be referring to that graph as well. Also on that graph, notice that I've shown a threshold potential. Okay, so let's go through the answer. It'll be the same as before. An animation will be shown, and then I will give you the marking point. So sodium ion concentration increases in the axon from an adjacent depolarisation. So the depolarisation will have happened in the adjacent part of the neuron, and sodium ions are diffusing from that region. Because the membrane potential is becoming less negative because of those sodium ions, some, not all, some voltage-gated sodium ion channels open. Sodium ions can then diffuse down their gradient or concentration gradient into the axon. And on the graph, we can see that. So we get this increase in membrane potential becoming less negative. So let's continue with the animation. So if that threshold potential is reached, then we get many voltage-gated sodium channels opening, and we get an influx of sodium ions down their electrochemical or concentration gradient into the axon. And we can see that on the graph with an increase in membrane potential now becoming positive. That means we've reached an action potential. So the final marking point there, you're referring to the fact that the inside is now positive relative to the outside. The membrane is said to be depolarized. Okay, so now on to membrane repolarization. So you can mark your answer on this one. So we're starting from that final point from the last question, which is we have membrane depolarization. How do we get back to that resting potential? So we'll be using the graph again on the right-hand side of the screen, and we'll do as we've done before. The animation will go through, and then we'll go through each marking point as we get to it. So when we reach that action potential, it can vary, but we're going to refer to a plus 40 millivolts here. Voltage-gated sodium ions channels close. And then we've got those voltage-gated potassium ion channels in the axon or neuron membrane. They open. Potassium ions diffuse out of the axon. Really important you get the direction of diffusion of ions correct. And you can see on the graph there that the membrane potential now drops to become negative again. Let's continue. So you notice on the graph that the membrane potential now drops below the resting potential. The membrane is said to be hyperpolarized, and that's because the potassium ion channels are slow to close, so an excess of potassium ions can diffuse out. And then finally, see on the graph there, we get back to resting potential, and that is because, as what we know, that resting potential is established because of the sodium-potassium pump and those leaky potassium ion channels. Okay, so hopefully you got all the answers correct for that. Okay, let's move on to the next question. So now describe the all or nothing response. So your answer should be this. And we can look at that on a graph. So we can take a look at a graph here. We've got the axon membrane potential on the y-axis, x-axis we've got time, and then I'm showing you a threshold potential there, somewhere around minus 55. But as always, you have to be flexible with what information you're given for questions. So if we look at an example here, this has not reached the threshold, this stimulus, and so we get no action potential. We get nothing. But in this case, threshold has been reached and we get an action potential. That is the all or nothing response. Okay, question 5. So just before we get into the answer to this, I'm just going to have a quick discussion about an unmyelinated neuron and how that works. So we'll discuss that first, and then we'll go into the answer for question 5. So let's take a look at an example of a sensory neuron, an unmyelinated sensory neuron. And at one end of this sensory neuron, we have a Pacinian corpuscle, when you should know how that functions and works. Let's imagine that we apply pressure to that Pacinian corpuscle and we end up with an increase in sodium ions in the sensory neuron ending because of those stretch-mediated sodium ion channels opening. And those sodium ions will diffuse from that sensory neuron ending along into the axon. And if we look at the axon, and we take a look at what effect that has, what it's going to do is have an effect on the membrane potential. So I'm going to plot a graph, and we're going to look at each section and how it affects each section of that axon. So in that first part, as those sodium ions diffuse in, they're going to increase the membrane potential. They're going to make it less negative, and we know about that. We've explained it in the previous answers. And then obviously, if it reaches threshold, if the stimulus is strong enough, then we get an action potential. And then there will be repolarization of that region of the axon. And then sodium ions from that action potential will diffuse further along the axon, and they will bring about the same effect in the next region of the axon. So we get the same effect. We get an action potential in that region. And then those sodium ions diffuse along, and they have, provided there are enough sodium ions that reach threshold potential, then that continues and continues and continues. That is how an action potential is propagated along an unmyelinated neuron. So section by section, we get this action potential. So now let's go to a myelinated neuron and go to the answer for question 5. And obviously the arrows just appeared there to show the direction of the progression of that impulse. Now let's go to the myelinated neuron and the answer to question 5. So in the case of a myelinated neuron, it propagates along by a process of saltatory conduction. So this is the answer. And that is as a consequence of these myelin sheaths and those action potential occurring between the gaps between myelin sheaths, called the nodes of Ranvier. And so we can take a look at a diagram of this process. And of course, the explanation is really that, or the importance of this is that the impulse travels along the myelinated neuron much faster than it would do in a non-myelinated neuron. So now let's have a look at the diagram. We've got a myelinated neuron here. We'll take a look at a section of that myelinated neuron. We use a similar diagram to what we showed with the unmyelinated neuron, but here now we're showing the myelin sheaths, and in between those myelin sheaths we have the nodes of Ranvier. And so what would happen is when we get an action potential occurring, when we get those sodium ions diffusing in, we would get an action potential only at that node of Ranvier. And then those sodium ions would diffuse along. They're unable to diffuse out, or any ions are unable to diffuse across the membrane where the myelin sheath is. So the only place we can get another action potential is at the next node of Ranvier, and so on and so on. And so as a consequence, this impulse is almost jumping, hence the term saltatory, saltation meaning jumping, between the nodes of Ranvier along the motor neuron, and that speeds up the rate of transmission of that impulse. Okay, question 6. What is the refractory period? Explain its importance. So the refractory period is this. So there's a short period of time where the voltage-gated sodium ion channels can't open, they are deactivated. And we can show that on the graph of an impulse, and it's the purple shaded area here. That is the refractory period. From the peak of the action potential when the membrane is depolarized to the early phase of repolarization, or that resting potential. What about its importance, though? There are 3 key reasons you need to explain its importance. So 1, ensures action potentials only move in one direction. So if we look at this on this graph, we've got 4 action potentials, and they can only go to the right. They can only move in that direction. They cannot go backwards, because an action potential cannot occur after a previous one has occurred. And if we consider that in the context of a neuron, then in the case of this sensory neuron with the pacinian corpuscle, it will only move away from the pacinian corpuscle towards the central nervous system, which is important because we wouldn't want that impulse to travel back towards the Pacinian corpuscle because it would not lead to any sort of response. Secondly, it ensures the action potentials are separated, so we don't get this overlapping of action potentials. So on the diagram below, I've shown if the refractory period didn't occur, then we could get this overlapping, and that would create quite a confused signal, unable to be interpreted by the brain, by the central nervous system. So by having that refractory period, what we get is this. We get these discrete impulses that can be interpreted by the brain. And finally, the refractory period limits the number of action potentials. So it limits the strength of a stimulus. So here we're showing the maximum number of impulses that could occur in this case. Okay, and finally to question 7. So you should know 3 factors that affect the speed of impulse transmission, and they are obviously temperature. You should always be thinking about kinetic energy. So as temperature increases, it's going to increase the kinetic energy of ions and the rate at which they diffuse. And it's also going to affect enzyme function. So it's going to affect the kinetic energy of the substrates and enzymes of respiration, the successful collisions between substrate and enzyme, and affect the rate of that process. But obviously remember, there is a limit. If temperature goes beyond the optimum temperature of those enzymes, they will lead to denaturation, and then it would obviously reduce impulse transmission. Similarly, with the lower the temperature, the slower the impulse transmission because of a lowered kinetic energy of ions and substrates and enzymes. 2, diameter of the axon. So the larger the diameter, the fewer ions leak, and so the greater the speed of impulse transmission. So faster impulses along large diameter axons. And finally, as you know, myelination. So the speed of transmission along myelinated axons is greater than unmyelinated axons. Okay, so that's all the information you need on nerve impulses. Repeat the questions as many times as you need to for you to understand it and have a strong knowledge of this topic.